Prokaryotic cell is the one with no well-defined nucleus present in it, while the eukaryotes have a well-defined nucleus in them with a nuclear membrane surrounding it. Nuclear envelope is absent in prokaryotes and present in eukaryotes. Ribosomes present in prokaryotic cell are of 70s, with smaller subunit of 30s and larger subunit of 50s. Ribosomes present in eukaryotic cell are of 80s, with smaller subunit of 40s and larger subunit of 60s. DNA of prokaryotes is circular and closed, while eukaryotes have one linear DNA, which is the genetic material present in the nucleus, and the other is circular present in mitochondria, called as mitogenome. Histone proteins are absent in prokaryotes, but present in eukaryotes. Despite absence of histone proteins in bacteria, bacterial chromosomes are present. Prokaryotes have chromatin, which is a DNA without histone protein. Eukaryotes' chromosomes varies from species to species in size and shape. First chromosome is the largest and 23rd is the smallest. Based on the shape of chromosomes, humans have chromosomes of shape I, J, and V. Glycocalyx is a combination of slime layer and capsule present in prokaryotes, but absent in eukaryotes. Cell wall of prokaryotes, mainly bacteria, is made of peptidoglycan, and of eukaryotic plant contains cellulose, chitin in fungi, and cellulose in algae. Plasma membrane of prokaryotes lacks sterols, while cholesterol is present in cell membrane of eukaryotes. Prokaryotes lack cytoskeleton, while a well-developed cytoskeleton is present in eukaryotes. No cyclosis is observed in prokaryotes, while it is observed in eukaryotes due to presence of cytoskeleton. Cell division occurs through binary fission in prokaryotes, while in eukaryotes mitosis and meiosis characterize the cell division. Sexual reproduction is not directly observed in prokaryotes as there is no gamete formation. However, it occurs through transduction, transformation, and conjugation. Eukaryotes involve sexual reproduction, where gamete formation occurs clearly in the meiosis process. Plasmid is an extrachromosomal circular DNA, which is only present in prokaryotes. Transcription in prokaryotes takes place in cytoplasm. In eukaryotes, transcription can take place in nucleus, mitochondria, and chloroplast. There are no post-transcriptional modifications in prokaryotes. In eukaryotes, addition of cap at 5' end and addition of tail at 3' end occurs. Translation occurs in cytoplasm in case of prokaryotes, while in eukaryotes it occurs in cytoplasm in association with rough endoplasmic reticulum and can take place in mitochondria and chloroplast as well. RNA polymerase of prokaryotes is of single type, and it encodes for all 3 RNA polymerases. Eukaryotic RNA polymerases are 3 in number. RNA polymerase 1 encodes for ribosomal RNA, RNA polymerase 2 encodes for messenger RNA, and RNA polymerase 3 encodes for transfer RNA molecule. Replication site in prokaryotes has a single origin, while eukaryotes have multiple origin of replication as the size of genomes is very large. The length of Okazaki fragments in prokaryotes is 1,000 to 2,000 nucleotides, while eukaryotic Okazaki fragments are 100 to 200 nucleotides long. There is no division of labor in prokaryotes, but eukaryotes do have division of labor and compartmentalization. Mesosomes are present in the cell membrane of prokaryotes, while eukaryotes have no mesosome system. Mitotic apparatus is not formed in prokaryotic cells, it only occurs in eukaryotic cells. The prokaryotic cells can vary in size only up to micrometers, while eukaryotes range from few micrometers to centimeters. The cellular organization in prokaryotes is only unicellular, while eukaryotes are unicellular, for example yeast, and multicellular as plants and animals. Photosynthetic prokaryotes use hydrogen sulfide, nitrate, sulfate, and oxygen as molecules, while eukaryotic photosynthesis only utilizes oxygen and water molecules. Prokaryotes have bacterial chlorophyll in them, and eukaryotes have 3 different chlorophyll, such as chlorophyll A, B, and C. In formation of initiation complex, eukaryotes small subunit of 30s is involved, while in eukaryotes smaller subunit of 40s is involved. Elongation process of protein translation in prokaryotes is carried out by larger subunit of 50S, and in eukaryotes elongation is carried out by 60S subunit of ribosomes. Prokaryotic promoters are -35 and -10, while eukaryotic promoters are -75 and -25. From evolutionary point of view, prokaryotes are more primitive form of life, while eukaryotes are less primitive. Vacuoles is absent in prokaryotes and present in eukaryotes.